Right, and now proceeding from there, how are tumor markers used in cancer care? Right, welcome everybody. We're back with the lectures. I'm Dr. Bobby on behalf of New Age Cosmetic White EdTech Healthcare Consultancy. I welcome all aspirants for the qualifiers to the various exams of NEET that require biology as one among the basics that need to be cleared. For example, the NEET exams, the All Institute of Medical Sciences, and the Council of Scientific and Industrial Research that tests your ability through the National Eligibility Test. Welcome back. On to a continuation of the subject of cancer, we have the next question to be taken up. So the question is, how are tumor markers used in cancer care? Tumor markers may provide a wide variety of information that is important for cancer care. And as we proceed, let's remember to thank the National Cancer Institute and the United States government and the Federal Drug Administration for having given us so much of access to details to study from as students of medicine, dentistry, and paramedical sciences, and all those of you appearing for the exams to make it as healthcare professionals. Once again, credits to NCI and all associations associated with them. Thank you. And tumor markers can provide a wide variety of information that is important for cancer care, such as helping to diagnose cancer. However, having an elevated level of tumor marker does not mean that someone has cancer. Non-cancerous conditions can sometimes cause an increase in the level of a tumor marker. In addition, not everyone with a particular type of cancer will have a high level of tumor marker associated with that cancer. Therefore, measurements of tumor markers are usually combined with the results of other tests, such as biopsies or imaging, to diagnose cancer. The type of cancer, the stage of cancer, an estimate of prognosis, what type of treatment may be effective, tumor markers that indicate whether someone is a candidate for a particular targeted therapy are sometimes referred to as biomarkers for cancer treatment. Biomarkers are generally measured in samples of tumor tissue. However, tumors can shed cells or bits of biological material into blood, and these can be measured by tests called liquid biopsies. Well, let's look into biomarkers for cancer treatment. Right. Biomarker testing for cancer treatment. What is a biomarker testing for cancer treatment? Right, we just read through, but we're just going back again to the same thing, just to freshen up how well we are getting exposed to the subject. So, as freshers, you need to have multiple exposures to the subject. Keep on reading to get the details right, and then to understand what it is all about. Now, biomarker testing is a way to look for genes, proteins, and other substances called biomarkers or tumor markers that can provide information about cancer. Each person's cancer has a unique pattern of biomarkers. Some biomarkers affect how certain cancer treatments work. Biomarker testing may help you and your doctor choose a cancer treatment for you. Right? There are also other kinds of biomarkers that can help doctors diagnose and monitor cancer during the treatment. To learn more about this is the tumor markers fact sheet. Right? So what's that? Well, we just read through that. That portion is just about clear when we read it. Tumor markers and about the body fluids. Okay, how are tumor markers used in cancer care? Tumor markers can provide a wide variety of information that is important for cancer care, such as helping to diagnose all those details, which also we had read. Now, continuing with biomarkers. Biomarkers are generally measured in samples of tumor tissue. However, tumors can shed cells or bits of biological material into blood, and these can be measured by tests called liquid biopsies. Right? We just read through that. How well the treatment is working. Periodic or serial measurements of a marker made while someone is undergoing treatment can indicate whether the tumor is responding to treatment, whether cancer has returned measuring tumor markers periodically after treatment has ended, may be used to check for recurrence. And if you have questions, connect with the cancer information specialist on the details below. You have phone 1-800-4-CANCER, chat live help, email is at nciinfo@nih.government. Available from Monday to Friday, 9 a.m. to 9 p.m. Eastern Time. All right, what tumor markers tests are currently being used and for which cancer types? A number of tumor marker tests are currently being used for a wide range of cancer types. See the list of tumor marker tests in common use for more information. Let's go into that. Tumor marker tests in common use. A tumor marker is anything present in or produced by cancer cells and other cells of the body in response to cancer, certain drugs, non-cancerous conditions that provides information about a cancer, which was just read, such as how aggressive it is. I'm sure that repetitive reading of certain portions will help you understand what exactly the whole subject is about for those who are fresh into the field, right? So just listen carefully, make notes, so that what you don't understand, you can reread on them, right? You need to get to know the basics if you want to go into the advanced portions. So that's how it's happening. So that's why we enumerated the cells about cell biology, cell signaling, cell communications, and all that, right? Okay. I think we've seen this tumor markers page for information regarding the targeted therapy or whether it is responding to treatment. Continuing from there, listed below are tumor markers tests that are in common use and maintained mainly to determine treatment or to help make a cancer diagnosis of cancer or a cancer diagnosis. This list is not comprehensive. In particular, tumor markers frequently become available and may not be included here. The list also does not include the many tumor tests that are tested by immunophenotyping and immunohistochemistry. Know these terminologies and get to familiarize yourself with these terminologies, right? Okay. And the many tumor markers that are tested by immunophenotyping and immunohistochemistry to help diagnose cancer and to distinguish between different types of cancer. The tumor markers listed below are targets for targeted therapy in multiple cancers, but serve as tumor markers for only a subset of cancers. For each set, the main cancer types that it is used for are listed. Some markers may be used for other cancer types that are not listed. ALK, gene arrangements, and overexpression. Now, cancer types are cancer-like conditions, non-small cell lung cancer, anaplastic large cell lymphoma, histiocytosis. What's analyzed is a tumor. How used to help determine treatment and prognosis. Now, next is alpha-fetoprotein AFP. Right, cancer types: liver cancer, ovarian cancer, and germ cell tumors. What's analyzed? Blood. How used to help diagnose these cancers and follow response to treatment, to assess stage, prognosis, and response to treatment of germ cell tumors. B-cell immunoglobulin, gene arrangement, cancer type, B-cell lymphoma. What's analyzed? Blood, bone marrow, or tumor tissue. How it's used? To help in diagnosis, to evaluate effectiveness of treatment, and to check for recurrence. BCL2 gene arrangement. Cancer types: lymphomas, leukemias. What's analyzed? Blood, bone marrow, or tumor tissue. How used? For diagnosis and planning therapy. BCR-ABL fusion gene, Philadelphia chromosome. Now, I think you've heard of that. And we'll go into the details of the various tests. The cancer types would be chronic myeloid leukemia, acute lymphoblastic leukemia, and acute myelogenous leukemia. Now, what's analyzed? Blood or bone marrow. How used? To confirm diagnosis, predict response to targeted therapy, help determine treatment, and monitor disease status. Beta-2 microglobulin, B2M. Cancer types are multiple myeloma, chronic lymphocytic leukemia, and some lymphomas. What's analyzed? Blood, urine, or cerebrospinal fluid. How used? To determine prognosis and follow response to treatment. Next would be beta human chorionic gonadotrophin, beta HCG. You might have read these terms somewhere, so just get to know the basics here. Cancer types, carcinoma and germ cell tumors. What's analyzed? Urine or blood. How used? To assess stage, prognosis, and response to treatment. And the next would be bladder tumor antigen, BTA. Cancer types would be bladder cancer and cancer of the kidney or ureter. What's analyzed? Urine. How used? As surveillance with cytology and cytoscopy of patients already known to have bladder cancer. BRAF V600 mutations. Cancer types are cancer-like conditions: thyroid cancer, melanoma, ovarian cancer, Erdheim-Chester disease, Langerhans cell histiocytosis, hairy cell leukemia, colorectal cancer, non-small cell lung cancer, and other cancer types. What's analyzed is a tumor. How used? To help determine treatment. Next is BRCA1 and BRCA2 gene mutations we just read about as an instance of reference. Well, this has to do with breast cancer, ovarian cancer, pancreatic cancer, and prostate cancers that are checked using these gene mutations. What's analyzed? Blood and or the tumor. How used? To help determine treatment. So CA 15.3 to CA 27.29. So cancer type is breast cancer. What's analyzed? Blood. How it's used? To assess whether treatment is working or the cancer has recurred. CA 19-9. Cancer types would be pancreatic gallbladder, bile duct, and gastric cancers. What's analyzed is blood. How used? To assess whether treatment is working. Right? You have any questions? Here's the section. You can connect with the cancer specialists in the US over here and the Indian sector. If there's any requirement, you can let me know. We can get you the contact details of the various cancer research institutions like RCCA and so on. RCC is in Trivandrum. You have other centers throughout the country. So if you require any details, let me know. We can get you in touch with the NGOs and the cancer centers. Right? So proceeding to the next section is CA 125. For that, let me see if I missed anything out. No, I'm not. CA 125. Cancer type is ovarian cancer. What's analyzed is blood. How used? To help in diagnosis, assessment of response to treatment, evaluation of preference. CA 27.29. Cancer type is breast cancer. What's analyzed is blood. How used? To detect metastasis or recurrence. Next is calcitonin. Cancer type is medullary thyroid cancer. What's analyzed is blood. How used? To aid in diagnosis, check whether treatment is working, and assess recurrence. The next will be carcinoembryonic antigen, CEA. Cancer types will be colorectal cancer and some other cancers. What's analyzed is blood. How used? To check effective treatment and detect recurrence. CD19. Cancer types will be B-cell lymphomas and leukemias. What's analyzed? Blood and bone marrow. How used? To help in diagnosis and to help determine treatment. CD20. Cancer type, non-Hodgkin lymphoma. What's analyzed is blood. How used? To help determine treatment. CD22. Cancer types, B-cell lymphomas and leukemias. What's analyzed? Blood and bone marrow. How used? To help in diagnosis and to help determine treatment. CD25. Cancer type, non-Hodgkin T-cell lymphoma. What's analyzed is blood. How used? To help determine treatment. CD30. Cancer types, classic Hodgkin lymphoma, B-cell and T-cell lymphomas. What's analyzed is tumor. How used? To help determine treatment. CD33. Cancer type, acute myeloid leukemia. What's analyzed? Blood. How used? To help determine treatment. Next is granin A, CGA. Cancer type is neuroendocrine tumors. What's analyzed is blood. How used? Is to help in diagnosis, assessment of treatment response, and evaluation of recurrence. Chromosome 17p deletion, cancer type. Chronic lymphocytic leukemia, what's analyzed? Blood. How used? To help in diagnosis and to determine treatment. Chromosomes 3, 7, 17, and 9p21. Cancer type, blood cancer, bladder cancer. Let me repeat, cancer type is bladder cancer, for which the chromosomes 3, 7, 17, and 9p21 are checked. It's analyzed through your urine. How used? To help in monitoring for tumor recurrence. Circulating tumor cells of epithelial origin, cell search, right? Cancer types are metastatic breast, prostate, and colorectal cancers. What's analyzed? Blood. How used? Is to inform clinical decision making and to assess prognosis. Next is the c-KIT, bar CD117, the cancer types: gastrointestinal stromal tumor, mucosal melanoma, acute myeloid leukemia, and mast cell disease. What's analyzed? Tumor, blood, or bone marrow. How used? To help in diagnosis and to help determine treatment. Cyclin D1, CCND1, gene arrangement or expression. Cancer types are lymphoma, myeloma, what's analyzed as tumor. How used? Is to help in diagnosis. Cytokeratin fragment 21-1, cancer type is lung cancer, what's analyzed as blood. How used? Is to help in monitoring for recurrence. Des-gamma carboxyprothrombin DCP, a cancer type is hepatocellular cancer, carcinoma, what's analyzed as blood. How used? Is to monitor the effectiveness of treatment and to detect recurrence. DPD gene mutation, cancer types: breast, colorectal, gastric, and pancreatic cancers. What's analyzed is blood. How used? Is to predict the risk of a toxin reaction to 5-fluorouracil therapy. EGFR, cancer types: non-small cell lung cancer and colorectal cancer. What's analyzed? Tumor. How used? To help determine treatment and prognosis. ESR1 gene mutation, cancer type is breast cancer. What's analyzed as tumor. How it's used is to help determine treatment. Right, we proceed from ESR1 gene mutation. The cancer type is breast cancer. What's analyzed as tumor. How used is to help determine treatment. The next will be estrogen receptor, ER, or progesterone receptor, PR. Cancer type will be breast cancer. What's analyzed will be tumor. How used? To help determine treatment. FGFR2 and FGFR3 gene mutations. Cancer types would be bladder cancer and cholangiocarcinoma. What's analyzed as a tumor. How used would be to help determine treatment. Fibrin or fibrinogen. Cancer type is bladder cancer. What's analyzed as urine. How used? To monitor progression in response to treatment. Next is 5-HIAA. His cancer type is carcinoma tumors. What's analyzed as urine. And how used? To help in diagnosis and to monitor the disease. Next will be protein signature. 5-protein signature. OVA1 cancer type, ovarian cancer. What's analyzed? Blood. How used? To preoperatively assess pelvic mass for suspected ovarian cancer. And FTL3 gene mutations, cancer type would be acute myeloid leukemia. What's analyzed is blood. How used? To help determine treatment and prognosis. FO6 gene signature, prolaris. Cancer type would be prostate cancer. What's analyzed would be tumor. How used? It is to predict the aggressiveness of prostate cancer and to help manage treatment. Foundation 1 CDX F1 CDX genomic test. This cancer type is any solid tumor. What's analyzed is tumor. How used? As a companion diagnostic test to determine the appropriateness of treatment with specific targeted therapies and for general tumor mutation profiling. Foundation 1 liquid CDX. Cancer type, any solid tumor. What's analyzed is blood. How used? As a companion diagnostic test to determine the appropriateness of treatment with specific targeted therapies and for general tumor mutation profiling. Gastrin. Cancer type is gastrin-producing tumor, gastrinoma. What's analyzed is blood. How used to help in diagnosis, to monitor the effectiveness of treatment, and to detect recurrence. Guardant 360 CDX genomic test. Cancer type would be any solid tumor. What's analyzed is blood. How used as a companion diagnostic test to determine treatment and for general tumor mutation profiling. HE4 is cancer type ovarian cancer. What's analyzed is blood. How used is to plan cancer treatment, assess disease progression, and monitor for recurrence. HER2 by NEU, ERBB2, gene amplification, mutations, protein overexpression. Its cancer types will be breast, ovarian, bladder, pancreatic, non-small cell lung, gastroesophageal, and stomach cancers. What's analyzed is tumor. How used is to help determine treatment. IDH1 and IDH2 gene mutations. Cancer types: acute myeloid leukemia, cholangiocarcinoma, and glioma. What's analyzed? Bone marrow, tissue or blood. How used to help determine treatment. I think we owe a lot of credit to the NCI for so much of detail, awareness on how to proceed in any aspect of anybody. If, God forbid, such diseases come to anyone, but if in case anyone's suffering, this is a load of information. It's a beautiful ocean of information, which is so well informative and so helpful to so many candidates. So please listen carefully, take notes, and let it help you all to become good professionals, right? Okay, proceeding with the next one, immunoglobulins. Cancer types: multiple myeloma and Waldenstrom macroglobulinemia. What's analyzed? Blood and urine. And how used? To help diagnose disease and assess response to treatment and look for recurrence. The IRF4 gene rearrangement. Cancer type is lymphoma. What's analyzed? Tumor. How used? And to help in diagnosis. JAK2 gene mutation. Cancer type: certain types of leukemia. What's analyzed? Blood and bone marrow. How used? To help in diagnosis. KRAS gene mutation. Cancer types: colorectal cancer and non-small cell lung cancer. What's analyzed? Tumor, plasma. How used? To help determine treatment. Lactate dehydrogenase. Cancer types: germ cell tumors, lymphoma, leukemia, melanoma, and neuroblastoma. What's analyzed is blood. How used? To assess stage, prognosis, and response to treatment. Microsatellite instability, MSI, and or mismatch repair deficiency, DMMR. Cancer types: colorectal cancer, endometrial carcinoma, and other solid tumors. What's analyzed as tumor. How used? To guide treatment and to identify those at high risk of having inherited certain cancer predisposing syndromes. MYC gene expression. Cancer types: lymphomas, leukemias. What's analyzed is tumor. How used? To help in diagnosis, and to help determine treatment. MYD88 gene mutation. Cancer types: lymphoma, Waldenstrom macroglobulinemia. What's analyzed is tumor. How used is to help in diagnosis and to help determine treatment. Myeloperoxidase MPO. Cancer type is leukemia. What's analyzed is blood. How used? To help in diagnosis. Neuron-specific analysis, NSE. Cancer types: small cell lung cancer and neuroblastoma. What's analyzed as blood. How used? To help in diagnosis and to assess response to treatment. NTRK gene fusion. Cancer type: any solid tumor. What's analyzed is tumor. How used? To help determine treatment. And the next would be nucleomatrix protein 22. Cancer type will be bladder cancer. What's analyzed is urine. How used is to monitor response to treatment. PCA3 mRNA. Cancer type is prostate cancer. What's analyzed is urine collected after digital rectal exam. How used? To determine need for repeat biopsy after negative biopsy. PIK3CA gene mutation status. Cancer types: breast cancer, colorectal cancer, non-small cell lung cancer, and ovarian cancer. What's analyzed as tumor. How used? To assess the likely cause of the disease, prognosis, and to determine the appropriateness of treatment with specific therapies. The next would be PML, RAR alpha fusion gene. Cancer type, acute promyelocytic leukemia, APL. What's analyzed is blood and bone marrow. How used? To diagnose APL, to predict response to all-trans retinoic acid or arsenic trioxide therapy, to assess effectiveness of therapy, to monitor minimal residual disease, and to predict early relapse. Programmed death ligand 1, PDL1. Cancer types, non-small lung cancer, triple negative breast cancer, and liver cancer, stomach cancer, gastroesophageal junction cancer, cervical cancer, bladder cancer, head and neck cancer, and classical Hodgkin lymphoma and other aggressive lymphomas subtypes. What's analyzed? A tumor. How used? To help determine treatment. Prostate-specific antigen, PSA. Cancer type, prostate cancer. What's analyzed? Blood. How used? To help in diagnosis, to assess response to treatment, and to look for recurrence. Prostatic acid phosphatase, BAP. Cancer type, metastatic prostate cancer. What's analyzed? Blood. How used? To help in diagnosing poorly differentiated carcinomas. RAT gene fusions and mutations. Cancer type, medullary thyroid cancer, non-small lung cancer, non-small cell lung cancer. What's analyzed? Tumor. How used? To help determine treatment. ROS1 gene arrangement. Cancer type, non-small cell lung cancer. What's analyzed? Tumor. How used? To help determine treatment. 17 gene signature oncotype DX GPS test. Cancer type, prostate cancer. What's analyzed? Tumor. How used? To predict the aggressiveness of prostate cancer to help manage treatment. 70 gene signature mammaprint. That's the cancer type is for breast cancer. What's analyzed is tumor. How used? To evaluate the risk of recurrence. Next will be soluble mesothelin related peptide SMRP. Cancer type is mesothelioma. What's analyzed is blood. How used? To monitor progression or recurrence. Somatostatin receptor. Cancer type, neuroendocrine tumors affecting the pancreas or gastrointestinal tract, GEP-NETs. What's analyzed? Tumor by diagnostic imaging. How used? To help determine treatment. T-cell receptor gene rearrangement. Cancer type is T-cell lymphoma. What's analyzed? Bone marrow, tissue, example lymph node, and blood. How used? To help in diagnosis and to detect and evaluate residual disease. Terminal transferase, TDT. Cancer types, leukemia, lymphoma. What's analyzed is tumor or blood. How used? To help in diagnosis. Thiopurine S-methyltransferase, TPMT, enzyme activity or TPMT genetic tests. Cancer type is acute lymphoblastic leukemia. What's analyzed? Blood and buccal cheek swab. How used? To predict the risk of severe bone marrow toxicity. Myelosuppression with thiopurine treatment. Thyroglobulin, cancer type, thyroid cancer. What's analyzed is blood. How used? To evaluate response to treatment and to look for recurrence. TP53 gene mutations. Cancer type is B-cell chronic lymphocytic leukemia. What's analyzed is blood. How used? To help determine treatment and prognosis. Tumor mutational burden, TMB. Cancer type is blood solid tumors. What's analyzed as tumor. How used? Is to help determine treatment. 21 gene signature is oncotype DX. Cancer type is breast cancer. What's analyzed as tumor. How used? To evaluate the risk of distant recurrence and to help plan treatment. UGT1A1 asterisk 28 variant homozygosity. Cancer type is colorectal cancer. What's analyzed is blood and buccal cheek swab. How used? To predict toxicity from irinotecan therapy. urine catecholamines, VMA, and HVA. Cancer type is neuroblastoma. What's analyzed? Urine. How used is to help in diagnosis. Urokinase plasminogen activator, UPA, and plasminogen activator inhibitor, PAI1. Cancer type is breast cancer. What's analyzed is tumor. How used to determine aggressiveness of cancer and guide treatment. This is reviewed on December the 7th, 2023. I just read for them just in case you would like to reproduce some or all of this content, and reuse of NCI information for guidance about copyright and permissions. In case of permitted digital reproduction, please credit the National Cancer Institute as a source and link to the original NCI product using the original product title as example. Tumor marker test in common use was originally published by the National Cancer Institute. Thank you very much. Courtesy National Cancer Institute. And for further details, you can go for syndication services. You can also go to the digital platform. So that's it for this lecture, and we're continuing with the next portion. Hold on. All right, just for general info, unless and otherwise indicated, all text within NCI products is free of copyright and may be reused without our permission. Credit the National Cancer Institute as a source. In the case of digital reproduction, please link to the original NCI product using the original product's title. For example, targeted therapy to treat cancer was originally published by the National Cancer Institute. Consider linking directly to our content rather than reproducing it, as our content may be updated over time. Some of our content is also available for syndication. More about our syndication services. Reuse of graphics, photos, illustrations used in National Cancer Institute information products are a mix of copyrighted and copyright-free materials. If you want to reuse a graphic independently of the original NCI product, please follow this guidance. And graphics created and owned by private sector designers will be credited to individuals or companies. Permission to reuse these copyrighted graphics must be negotiated directly with the creators and not NCI. If you're not sure with who the creator is, email us at nci.info@nih.government. Well, I guess we are not going into too much of detail. We're just reading. We just want to know the basics, get to have more awareness, and be thankful to NCI for what they're doing for us. All right, and credit NCI always with these topics. Graphics explicitly credited to NCI are copyright free, so we just go by that. That's enough, and may be used without our permission. Please credit the National Cancer Institute as a source. Visit NCI Visuals online to find copyright free measures you may download, and use. Reuse of NCI logo and PDQ trademark. The NCI logo and PDQ trademark may only be used in reproductions of original, wholly unchanged NCI products. Reuse of patient education publications. If you want to copy or print NCI patient education publications in their entirety with no changes, you may freely do so without our permission. So patients are lucky that you have so much of an ocean of information for the good. However, if you want to change anything about the publication, you must indicate the material is adapted from the NCI, National Cancer Institute. Refrain from using the NCI logo on your adapted product. Okay. Seek permission from the creator of any copyrighted graphics that appear in the original NCI product. Okay. Translations of NCI products. We welcome translation of NCI products into languages other than English as long as the following criteria are met. If you want to reuse copyrighted graphics from original NCI product, you must get permission from the copyright owners. Translated product may not use the NCI logo or PDQ trademark. That's if it goes into other languages. We're not going there. We're not translating or whatever. Translated product must credit the National Cancer Institute and include the statement: the National Cancer Institute, NCI, does not endorse this translation, and no endorsement of NCI should be inferred. Yes. If you still have questions or would like to seek permission to reuse copyrighted or trademarked items in your own materials, please email us at nci.info@nih.government. Thank you. Reviewed on February 13th, 2024. Right, now we're on to the fundamentals of cancer. We just got to know about the various tests and the basics. In fact, just minor basics. Well, let's proceed with the fundamentals of cancer. How does cancer form? That's what you see here are normal cells and cancer cells. This here is a normal cell. This here is a cancer cell. Right? Now when we go around, cancer is a disease caused when cells divide uncontrollably and spread into surrounding tissues. So before we proceed any further, let's give all credits to the National Cancer Institute in the United States of America and thank the federal government for making such invaluable material available to the general public and to those who are less privileged to know more about treatment aspects and studies, and for students who are less privileged to study, and those who can make use of these lectures to sit the various qualifiers to pass the basics to become healthcare professionals by appearing for the NEET exams, so National Eligibility Entrance Tests, and the All India Institute of Medical Sciences, and to the Council of Scientific and Industrial Research Forum and institutions that need these clarifications and qualifications through the National Eligibility Tests and similar exams in India and overseas. Welcome aboard. This is material that's internationally acclaimed, internationally accredited, and which is all screened and well documented by the Federal Drug Administration, FDA, in the United States government. So please pay attention to each lecture. Kindly do not reproduce this for any commercial purposes. Please use it for the best of your education and your examination purposes. Study and let it be of use to your future professions. Thank you for listening, and we proceed with the lecture. Cancer is a disease caused when cells divide uncontrollably and spread into surrounding tissues. I'm Dr. Bobby, and I welcome all of you to my daily lectures on behalf of New Age Cosmetic White EdTech Healthcare Consultancy, and we proceed with the lectures. Presently, it's on cancer, as we have all the references from National Cancer Institute in the United States of America for proceeding ahead with the fundamentals of cancer. Now, we're just proceeding on with the various diagrams. This one, the first one is about the normal cells and cancerous cells. We go to the next. This would be the gene. What causes cancer? The gene or cancer is caused by changes to DNA. Most cancer causing DNA changes, right, occur in sections of DNA called genes. These changes are also called genetic changes. And go on to the next picture. Next illustration, is about a cancer cell. What are oncogenes and the oncogene, right? The DNA change can cause genes involved in normal cell growth to become oncogenes. Unlike normal genes, oncogenes cannot be turned off, so they cause uncontrolled cell growth, right? You've seen that. The next would be what are tumor suppressor genes? This is a normal cell and the other is a cancer cell. Tumor suppressor genes in normal cells prevent cancer, right? DNA changes that inactivate tumor suppressor genes can lead to uncontrolled cell growth. So when there's an absence of tumor suppressor genes, you have cancerous growth. So in normal cells, tumor suppressor genes prevent cancer by slowing or stopping cell growth. DNA changes that inactivate tumor suppressor genes can lead to uncontrolled cell growth and cancer. Next. This diagram has to do with what is a tumor microenvironment of a tumor. This is a T cell, myeloid cell, macrophage cell, a natural killer cell, a dendritic cell, regulatory T cell, fibroblast, and cancer cells, right? So within a tumor, cancer cells are surrounded by a variety of immune cells, fibroblasts, molecules, and blood vessels, what's known as the tumor microenvironment. So they ask you a question, what do you mean by tumor microenvironment? Remember to mention all these. Remember this diagram. If you can produce a diagram like this and write your answers, that's it. You'll top the exams. You can ace the exams. So be very attentive and make notes. Cancer cells can change their microenvironment, which in turn can affect how cancer grows and spreads. Next one. This is about the immune system cells that can detect and attack cancer cells, but some cancer cells can avoid detection or thwart an attack. Some cancer treatments can help the immune system better detect and kill cancer cells. So this is what it is. How does the immune system interact with cancer? This is the attack and that's the evade. So make notes of this, take a screenshot and try to draw these diagrams and learn from it, okay? Draw it yourself. If you can do that much, nothing like it. Okay, the next one. Each person's cancer has a unique combination of genetic changes. Specific genetic changes can make a person's cancer more or less likely to respond to certain treatments. So this shows, this diagram is how do genetic changes affect cancer treatment. The response and no response. Right? Next is about, this is what causes genetic changes. Hereditary, UV radiation, chemicals, viruses, smoking, and cell dividing. Now, genetic changes that cause cancer can be inherited or arise from certain environmental exposures. Genetic changes can also happen because of errors that occur as cells divide. Right? Next. Most often cancer-causing genetic changes accumulate slowly as a person ages, leading to a higher risk of cancer later in life. This is how does age relate to cancer. DNA change, the child, the adult, and the senior. Right? And the next, I think that will be the last slide shot for this section. So cancer cells can break away from the original tumor and unravel and travel through the blood or lymph system to distant locations in the body where they exit the vessels to form additional tumors. This is called metastasis. Right? Metastatic tumor. And here the blood vessel, cancer cell, blood cell, the original tumor. So that's it for these diagrams. Excuse me. Now we're going for the types of genes that cause cancer. The genetic changes that contribute to cancer tend to affect three main types of genes. Please make special notes for these. Proto-oncogenes, tumor suppressor genes, and DNA repair genes. These changes are sometimes called drivers of cancer. So the question comes, what are the drivers of cancer? The answer should be proto-oncogenes, tumor suppressor genes, and DNA repair genes. So proto-oncogenes are involved in normal cell growth and division. However, when these genes are altered in certain ways or are more active than normal, they may become cancer-causing genes or oncogenes, allowing cells to grow and survive when they should not. Tumor suppressor genes are also involved in controlling cell growth and division. Cells with certain alterations, tumor suppressor genes may divide in an uncontrolled manner. DNA repair genes are involved in fixing damaged DNA. Cells with mutations in these genes tend to develop additional mutations in other genes and their changes in their chromosomes, such as duplications and deletions of chromosome parts. Together, these mutations may cause the cells to become cancerous. As scientists have learned more about the molecular changes that lead to cancer, they have found that certain mutations commonly occur in many types of cancer. Now, there are many cancer treatments available that target gene mutations found in the cancer. A few of these treatments can be used by anyone with a cancer that has targeted mutation, no matter where the cancer started growing. So let's see target gene mutations found in cancer. Right, targeted therapy to treat a cancer. What is targeted therapy? What are the types of targeted therapy? Who is treated with targeted therapy? How does targeted therapy work against cancer? Are there drawbacks to targeted therapy? What are the side effects of targeted therapy? What can I expect when having targeted therapy? And where can I find out about clinical trials of targeted therapy? Okay, so what is targeted therapy? Targeted therapy is a type of cancer treatment that targets proteins that control how cancer cells grow, divide, and spread it. It is the foundation of precision medicine. As researchers learn more about the DNA changes in proteins that drive cancer, they are better able to design treatments that target these proteins. What are the types of targeted therapy? Most targeted therapies are either small molecule drugs or monoclonal antibodies. Small molecule drugs are small enough to enter cells easily, so they are used for targeting targets that are inside cells. Monoclonal antibodies, also known as therapeutic antibodies, are proteins produced in the lab. Right? These proteins are designed to attach to specific targets found in cancer cells. Some monoclonal antibodies mark cancer cells so that they will be better seen and destroyed by the immune system. Other monoclonal antibodies, right, directly stop cancer cells from growing or cause them to self-destruct. Still others carry toxins to cancer cells. Learn more about monoclonal antibodies. Now, how monoclonal antibodies treat cancer is shown as a video by the National Cancer Institute, so I wish to share that with you. Please watch and understand and make notes.
Block, flag, deliver. How monoclonal antibodies treat cancer. Antibodies are molecules that our bodies make to help fight germs. Monoclonal antibodies are similar molecules that are made in laboratories and are used by doctors to find or treat cancer and other diseases. This video explains what monoclonal antibodies are and a few ways they're used to treat cancer. Antibodies are Y-shaped molecules that attach tightly to a target. They're very specific, meaning that each antibody attaches to only one target. An antibody and its target fit together like pieces of a puzzle. In the laboratory, scientists can make many identical copies of a monoclonal antibody that can attach to a specific target, such as a molecule on the surface of cancer cells. These monoclonal antibodies can block molecules cancer cells need to grow, flag cancer cells for destruction by the body's immune system, or deliver harmful substances to cancer cells. This makes them a valuable type of targeted therapy for treating cancer. For example, a monoclonal antibody called trastuzumab attaches to a molecule called HER2 on the surface of some cancer cells. Blocking HER2 keeps it from sending signals the cancer cells need to grow. Another example involves VEGF, which is a molecule that makes blood vessels grow. A monoclonal antibody called bevacizumab blocks VEGF. Blocking VEGF stops the growth of new blood vessels that the tumor needs to survive. A third example is the monoclonal antibody pembrolizumab. Pembrolizumab attaches to molecules called immune checkpoints on immune cells. Blocking immune checkpoints helps the immune cells kill cancer cells. Other monoclonal antibodies treat cancer by flagging cancer cells for destruction. For example, when the monoclonal antibody rituximab attaches to a molecule called CD20 on cancer cells, it acts like a flag for immune cells. The immune system sees this flag and destroys the cancer cells. Some monoclonal antibodies fight cancer by delivering drugs, toxins, or radioactive particles to cancer cells. For example, brentuximab vedotin is a monoclonal antibody that is linked to a chemotherapy drug. When the antibody attaches to its target on cancer cells, it delivers the chemotherapy drug, which kills them. Cancer researchers are continuing to investigate new ways to use the precision of monoclonal antibodies to treat cancer patients. For more information, see cancer.gov/MAB. Artwork and animation by Therese Winslow, LLC, and Devon Medical Art, LLC. Health and Human Services, National Institutes of Health, National Cancer Institute, cancer.gov, 1-800-4-CANCER. Produced June 2020.
So I guess you've just watched that video to learn how monoclonal antibodies such as trastuzumab, pembrolizumab, and rituximab are used to treat cancer. I hope I've pronounced that right. I hope it's right. It's pembrolizumab and rituximab are used to treat cancer. Now, who is treated with targeted therapy? Well, for some types of cancer, such as chronic myelogenous leukemia, also known as CML. Most people with that cancer will have a target for a certain drug so that they can be treated with that drug. Most of the time, your tumor wouldn't need to be tested to see if it contains targets for which there is a drug. Testing your cancer for targets that could help choose your treatment is called biomarker testing. See biomarker testing for cancer treatment. For more information, I guess we've already read through that and seen a portion of biomarker testing here on the screens. Right, you may need to have a biopsy for biomarker. I think you heard that too, with regard to biomarker testing. Biopsy is a procedure in which a doctor removes a piece of the tumor for testing. There are some risks to having a biopsy. These risks vary depending on the size of the tumor and where it is located. Your doctor will explain the risks of having a biopsy for your type of tumor. Look up your type of cancer on the list of targeted therapy drugs approved to treat specific cancers. I think we've seen the whole list to learn more drugs that can be an option for you. List of targeted therapy drugs. We could just have a quick look just in case. Or even otherwise, as we're learning the subject. A list of targeted therapy drugs approved by specific types of cancer. List of targeted therapy drugs approved for specific types of cancer. The FDA has approved targeted therapy drugs for the treatment of some people with the following types of cancer. Some targeted therapy drugs are listed more than once because they have been approved to treat more than one type of cancer. The generic drug name is listed first with a brand name in parentheses. Right? Targeted therapy approved for bladder cancer will be all these. I think we have a full list. Target therapy for brain cancer, for breast cancer, for cervical cancer, for colorectal cancer. See, I think we read about this aflibercept, so Zeltrap. Just trying to make you remember things. Target therapy approved for dermatofibrosarcoma protuberance. Imatinib mesylate or Gleevec is so this imatinib mesylate is the generic name. Gleevec is the brand name. Okay. Targeted therapy approved for endocrine, neuroendocrine tumors. Target therapy approved for endometrial cancer, for esophageal cancer. Therapy for head and neck cancer. Therapy for GIT, stromal tumor. Approved for giant cell tumor, for kidney cancer, and for leukemia. And that's a lot for leukemia. And then for liver and bile duct cancer. We're just about winding up this lecture, and we'll be continuing additional portions in the next lecture. So please be attentive and please stay tuned, right? And we'll give it as edition 1, 2, and 3 likewise. Lectures 1, 2, and 3. Keep following as I give you constant and daily lectures on various topics for the NEET and AIIMS exams and CSIR-NET exams too, right? Targeted therapy for liver and bile duct cancer, right? And targeted therapy approved for bile lung cancer. And for lymphoma, targeted therapy for malignant mesothelioma, for multiple myeloma, for myelodysplastic and myeloproliferative disorders, approved for neuroblastoma, approved therapy for variant epithelial fallopian tube and primary peritoneal cancers, pancreatic cancers, right? Plexiform neurofibroma, prostate cancer, skin cancer, soft tissue sarcoma, solid tumors anywhere in the body, stomach gastric cancer, systemic mastocytosis, thyroid cancer, and all this was updated on 22nd of August 2024. And this particular frame of the recording will be over in about a minute or so. And on behalf of New Age Cosmetic White EdTech Healthcare Consultancy, I welcome you to the ensuing continuous lectures. Please make notes take screenshots, brace yourself, and get ready for your exams. Be well prepared. Understand the basics. If you have any doubts, you're not sure of anything, you want to ask queries, or you want clarifications, please write to me. Put it on the comment section of the YouTube or the concerned lecture. You can also connect with me on my WhatsApp page. My number, which is international roaming, is 9188661251. I'm Dr. Bobby Jacob KORAH. I'm signing off on this particular lecture. I will meet you at the next lecture right on with the continuous portions on cancer as of today, right? So please remember these points that I've just talked to you. If you would like to reproduce some or all of these content, see reuse of NCI information for guidance about copyright and permissions, but presently there's no obstructions on no NOC is required. This is free to use, and I hope this is of use to all those looking for treatment.